Introduction to Endocrinology, the scientific study of hormones and endocrine glands. The endocrine system is structurally made up of small glands scattered throughout the body, such as the hypothalamus, pituitary, pineal gland, thyroid, parathyroid, adrenal glands, the pancreas, ovaries, and testes. There are also major organs that can act as endocrine glands when necessary, such as the heart, kidneys, and small intestines and stomach. Then there are tissues that have endocrine-like functions, such as the placenta and adipose. The endocrine system produces and secretes chemical messengers known as hormones via endocrine glands that influence metabolic activity in the body. Hormones play roles in regulating almost all body functions such as metabolism and energy balance, growth and development, water and electrolyte balance, nutrient balance of blood, reproduction, behavior, and mobilization of body defenses. There are several types of chemical messenger systems in the body. Endocrine hormones are just one of those systems. Endocrine hormones are produced and released by glands or specialized cells into circulating blood and act on tissues at another location. This is the key property that distinguishes an endocrine hormone from other chemical messengers. The fact that hormones are produced at one location and affect tissues at another location some distance away is the definition of a hormone. Neuroendocrine hormones are produced and released by neurons into circulating blood instead of at a synapse. Neuroendocrine hormones will travel in the blood to act on another tissue at another location. We'll see how neuroendocrine hormones work when we cover hypothalamic hormonal function. Neurotransmitters are chemical messengers that are produced and released by neurons and act locally at a synapse. We'll also learn how some chemical messengers are both endocrine hormones and neurotransmitters, depending on where they are produced and secreted. An example is norepinephrine. When norepinephrine is produced and secreted by the adrenal medulla gland, it acts as a hormone. When norepinephrine is produced and secreted by postganglionic sympathetic neurons at a synapse, it acts as a neurotransmitter. Paracrines are chemical messengers secreted by cells into interstitial fluid to act on neighboring cells. They don't travel in the bloodstream. Autocrines are chemical messengers secreted by cells into interstitial fluid to act on the same cell. They too do not travel in the blood. Cytokines are a broad group of chemical messengers that can act as autocrines and paracrines and or endocrine hormones. Types of glands. The endocrine system relies on endocrine glands. The body has two types of glands, endocrine and exocrine. Both are formed by epithelial tissue. The difference between the two is exocrine glands secrete their products into ducts that open up on the surface of the tissues where they are located, whereas endocrine glands secrete hormones directly into the interstitial fluid surrounding the gland. The general function of a hormone is to 1. Be secreted into extracellular fluid, 2. Travel through the bloodstream, 3. Bind with specific receptors on target cells, 4. Initiate reactions. Some hormones are designed to only affect the metabolic function of just one specific type of tissue, and some hormones are designed to affect the metabolic function of several tissues. Here I created a brief, non-comprehensive list of the difference between the two systems, between the nervous system and the endocrine system. What I want you to appreciate is that the main difference between the endocrine system and nervous system is the chemical messengers used, the mode of transportation, time, and distance. The endocrine system is slow-acting and long-lasting. Hormones travel in the blood to their target tissues, which can be some distance from their origin of their release. For example, growth hormone released by the anterior pituitary gland in the brain can act on the oppositional growth of the first metatarsal bone in the foot. In comparison, the nervous system is fast-acting and short-lived. Neurotransmitters travel down the axon to their target tissue, which is very close. Some chemical synapses are only 2 nanometers apart. Familiarize yourself with this list because you will be expected to compare and contrast these two systems. Classes of hormones. Hormones are separated into three classes. The largest class is proteins and polypeptides. These proteins and polypeptides are made from amino acids except tyrosine. The only difference between protein hormones and polypeptide hormones is just the length of their chain. They both possess the same properties. The next class is steroids, which are synthesized from cholesterol. In fact, all steroid hormones are synthesized from cholesterol. Now, the last class is the amines, which are synthesized from the amino acid tyrosine. The amines are further divided into two subclasses, the catecholamines and thyroid hormones, and we'll cover more of these subclasses in the next few slides. In addition to being the most abundant class of hormones, protein polypeptide hormones are produced in ribosomes, stored in vesicles, and secreted from endocrine glands or endocrine tissues by exocytosis. The ability of endocrine glands to store protein polypeptide hormones in their vesicles allows that gland to stockpile their hormones. Protein polypeptide hormones are water-soluble and can easily enter nearby capillaries. Because they are water-soluble, protein polypeptide hormones can travel freely through the bloodstream. That means they're not bound to a globular protein for transport. Protein polypeptide hormones bind with specific receptors on the target cell plasma membrane. Each hormone in the body has a specific receptor that it binds with. Therefore, hormones can only affect tissues that have the specific receptor for that specific hormone. Steroid hormones have a chemical structure similar to cholesterol, which makes sense. All amine hormones are synthesized from tyrosine. There are two subgroups of amine hormones, the catecholamines produced by the adrenal gland and thyroid hormones produced by thyroid glands. Each of these subgroups has very different properties. Amine hormones these thyroid hormone group of amine hormones are produced in the follicles of the thyroid gland. Again, thyroid hormones are synthesized from tyrosine. Basically, thyroid hormone is a long chain of tyrosine amino acids with 3 or 4 iodide ions attached to it. Before the iodide ions are attached to the long chain of tyrosine amino acids, it is called thyroglobulin. Thyroid hormones are lipid-soluble, just like steroid hormones. Now, these hormones can diffuse through the thyroid follicular epithelial cells into surrounding interstitial fluid, where they swiftly enter the nearby capillaries. Like steroid hormones, most of the quantity of thyroid hormone release must travel bound to a globular protein. The letters TBG you see on the diagram represent thyroid binding globulin. Only a small amount can travel free. Once the thyroid hormone reaches its target tissue, it will be released from the thyroid binding protein, and because it's lipid soluble, the thyroid hormone will be able to diffuse through the target cell's membrane. But it doesn't stop there. It'll continue through the cytoplasm to enter the target cell's nucleus, because thyroid hormone specific receptors are located within the nucleus of the target cell. On the other hand, the other amine group, the catecholamines, are produced in the adrenomedullary. These hormones, epinephrine and norepinephrine, are produced by chromaffin cells within the medulla. Catecholamines have very similar properties as those of proteins and polypeptide hormones. They are stored in vesicles, allowing the adrenal gland to stockpile, and they are released by exocytosis. And like polypeptide protein hormones, catecholamines are water-soluble, meaning they can travel through the bloodstream and act like protein polypeptide hormones. Norepinephrine and epinephrine bind with specific receptors located within the target cell's plasma membrane. The synthesis and release of most hormones are regulated by the negative feedback mechanism, meaning when serum concentrations of the hormones are high, it will inhibit further release of that hormone, whereas low serum levels of the hormone will stimulate the release of that hormone. Many hormones require a chain of events to stimulate release. We'll cover those chain of events when we cover the functions of the hypothalamus and the anterior pituitary gland. The positive feedback mechanism occurs when the biological action of a hormone causes additional secretion of the same hormone. The best example is uterine contractions to facilitate birth. Each contraction causes the baby's head to push against the mother's cervix. The greater the distension of the cervix stimulates the release of more oxytocin, which in turn causes greater contractile force, pushing the baby's head harder against the cervix, and so on, till the baby's head is delivered. Cyclic control of hormone secretion. The release of many hormones is controlled by our biological clocks. There are 3 regular clocks or cycles that hormones respond to. One is the circadian cycle, which releases specific hormones approximately every 24 hours. A good example of this release is growth hormones and cortisol. Another example is the ultradian cycle, which releases hormones more frequently than every 24 hours. And a good example of that is the release of insulin throughout the day in response to consuming carbohydrates. The last cycle I will mention is the infradian cycle, which releases hormones less frequently than every 24 hours. And a good example of that is the cycle that releases hormones that control a woman's menstrual cycle. Other factors involved in cyclic release of hormones, and those are developmental stages in life, such as infancy, childhood, adolescence, adulthood, and aging. These all affect when certain hormones are released in our body. One of the factors that determine the effectiveness of a hormone is the amount of the hormone produced and released. There are some pathological conditions we will cover in this course that are a direct result of abnormal amounts of certain hormones being released. Some conditions are a result of too much hormone being released, and some are a result of too little hormone being released. Timing is another factor that determines the effectiveness of a hormone. We talked about that in the previous slide. Later on in the course, we'll talk about how the timing of release of certain hormones is critical for the normal function of the female reproductive system. The time certain hormones are released during the day, week, or month is critical to maintaining homeostasis and responding to the variables in your daily life, such as when you eat or drink, or how you respond to stress. Transportation is another huge factor in determining the effectiveness of a hormone. Remember when I stated at the beginning of the lecture that the ability of a chemical messenger to travel through the vascular system is one of the properties that makes it a hormone? Well, what happens to the ability of a hormone to travel in the bloodstream if a patient has arteriosclerosis or extremely low blood pressure? Both of these conditions will affect the ability and or timing of when a hormone reaches its target tissue. Or what about binding to a transport protein? Remember, both steroids and thyroid hormones require binding to a transport globular protein to circulate in the bloodstream. There are conditions that affect the body's ability to produce adequate amount of these transport globular proteins, thereby affecting the effectiveness of those hormones. And then there are the receptors. Hormones can only cause an effective response on a target cell if the target cell has a healthy, normally functioning specific receptor for that hormone. Other factors that determine effectiveness of a hormone is the interaction of other hormones. For example, some hormones act as facilitators or inhibitors for other hormones. And then there is the activation and inactivation process that determines the effectiveness. Both of these processes involve several steps, and if there's a problem with any of the steps, it'll affect the effectiveness of the hormone involved. There are 3 types of stimuli that can cause a hormone to be released. One is a hormonal stimulus. Oh, that was wrong. I'm sorry. A humoral stimulus, where the hormone is released in response to blood levels of certain substances. A good example is when parathyroid hormone is released in response to low serum levels of calcium. A neural stimulus occurs in response to a nervous stimuli, such as when a preganglionic sympathetic neuron stimulates the adrenal medulla to release epinephrine and norepinephrine. The hormonal stimuli is the one we will cover most extensively in this course. A hormonal stimulus occurs when one hormone stimulates the release of another hormone. An example of this is the hypothalamus releases hormones that stimulate the anterior pituitary to release hormones. What are the mechanism of actions of hormones? 1. Binding to a receptor. 2. The second messenger system that stimulates intracellular enzymatic cascade. And 3. Activation of genetic material stimulating protein synthesis. We have already covered the fact that a hormone must first bind with a receptor to initiate any kind of response in a target tissue. No receptor means no response. We also talked about the location of hormone receptors, how some are located on the surface of a target cell's plasma membrane, some have intracellular locations such as the cytoplasm and the nucleus. Most surface hormone receptors located on cells' plasma membranes are G-protein linked receptors. G-protein linked receptors have 7 transmembrane segments that loop in and out of the cell's membrane, giving the appearance of a serpent. Therefore, this type of receptor is often referred to as serpentine. One part of the serpentine receptor that protrudes into the cell cytoplasm is coupled with a G-protein. This coupling typically occurs on the 5th arm of the 7-membrane transmembrane domain. G-proteins are made up of 3 parts, alpha, beta, and gamma, often referred to as trimeric proteins. Inactive G-proteins have a guanosine diphosphate molecule attached to the alpha subunit. Second messenger's mechanism of activation involves a relay between a protein or polypeptide class hormone, a receptor, a G-protein, an enzyme, and the second messengers produced by this mechanism. Most second messenger systems use G-protein coupled receptors to facilitate this mechanism of action. G-proteins are considered the molecular switch of the second messenger pathway system. There are several second messenger systems in the body. We will cover the 2 most common ones, the cyclic AMP second messenger system and the phospholipase C second messenger system. Starting with this diagram, I will describe the process of the cyclic AMP second messenger system. I need to point out that this is just a diagrammatic representation and not drawn fully accurate. For example, the 7-membrane serpentine G-protein link receptor on the left looks more like a cartoon femur, and the G-protein attached to it looks like a square puzzle piece instead of showing its alpha, beta, and gamma parts. But unfortunately, this is the best diagram I could find to demonstrate this action. I have the steps of this action written on the next slide, so you can just listen and observe and not worry about taking notes. In the cyclic AMP second messenger system, the first messenger is the hormone, which is represented by the pink circles on the left. The first action in this mechanism is the hormone binding with a specific plasma membrane G-protein receptor, represented by the purple femur-looking thing. The binding of a hormone to the serpentine receptor causes it to change shape, which activates the G-protein. The G-protein is activated by the exchange of a GDP molecule for a GTP molecule. This activation causes the G-protein's alpha subunit bound to the GDP molecule to separate from the beta and gamma subunits. G-proteins behave like a light switch. That's often why they're called the molecular switch. The switch is off or inactive when GDP is bound to the G-protein, and the switch is on when GTP is bound to the protein. Some G-proteins have stimulating properties while others have inhibitory properties. In this example, I'm describing the action of the stimulating G-protein. Next, the separated activated alpha subunit of the G protein moves along the plasma membrane on the inside of the cell until it finds an enzyme, adenylate cyclase. In this diagram, it shows the G protein moving towards the right to ultimately attach to another purple structure, which is representing the protein enzyme adenylate cyclase. Adenylate cyclase is considered the mediator of the hormone response second messenger system. As you can see here, the activated G protein will bind to the adenylate cyclase, stimulating the adenylate cyclase to convert ATP into cyclic AMP. The second messenger system is cyclic AMP. As long as the activated G protein is bound to adenylate cyclase, the enzyme will continue to generate cyclic AMP second messengers. Eventually, GDP will hydrolyze to GDP, thereby deactivating the G protein and stopping the production of cyclic AMP. There is an enzyme called cyclic AMP phosphatidase esterase that, if present, can inhibit the conversion of ATP to cyclic AMP. Cyclic AMP triggers a cascade of chemical reactions in the cell by activating protein kinase. Active protein kinase triggers a cascade of chemical reactions within the target cells, such as activating enzymes, stimulating protein synthesis, opening membrane ion channels, and stimulating cellular secretions, just to name a few. Cyclic AMP is rapidly degraded by an intracellular enzyme, therefore eliminating the need for controls to stop activity. So basically, this second messenger system makes the target cell do stuff. Now, if an inhibitory G protein were coupled to the 7-member transmembrane protein, the hormone receptor complex would inhibit adenylate cyclase, preventing the target cell from doing stuff. In other words, no reaction would occur. Now on to the phospholipase C second messenger system. In this diagram, I'll attempt to explain the phospholipase C messenger system. You will have probably noticed that I tend to switch between using the term mechanism and system when talking about how second messengers work. Either term is correct. In the phospholipase C mechanism, the first two steps are the same as in the cyclic AMP second messenger mechanism. The first messenger is the hormone, a protein polypeptide hormone to be specific. The first action is when the hormone binds to the 7-member transmembrane G protein receptor and causes the receptor to change shape. The change in shape activates the G protein. A G protein is activated by exchanging the GDP molecule for a GTP molecule. The G protein alpha subunit with a GTP molecule attached to it will separate from the beta and gamma subunits and travel along the plasma membrane until it finds the phospholipase C enzyme. When the G protein binds to the phospholipase C enzyme, it causes the enzyme to split a specific membrane phospholipid called phosphatidylinositol biphosphate. That's a mouthful, so I will refer to it as PIP2 from now on. The PIP2 splits into two second messengers called diacylglycerol, or DAG, DAG, and inositol triphosphate, or IP3. DAG activates protein kinase enzymes, which will stimulate the cell to do the same thing protein kinase does in the cyclic AMP system, such as activating enzymes, stimulating protein synthesis, opening membrane ion channels, and stimulating cellular secretions. IP, the other second messenger, releases calcium from intercellular storage sites. The calcium then binds with calmodulin, which allows and activates a cascade of enzymes, stimulating protein synthesis, and etc., and etc. And again, the job of the second messengers is to make the target cell do stuff. Intracellular cascade is just the big picture of intracellular enzymatic cascade that is a single hormone can stimulate its target cell to generate millions of a final product. Steroid hormone mechanism of action. Steroids mechanism of action is much more simple than that of the second messenger system used for protein polypeptide hormones. First, the lipid-soluble steroid hormone diffuses across the target cell's phospholipid membrane into the cytoplasm. Then, in the cytoplasm, it binds with a specific receptor protein. Then the steroid hormone receptor complex enters the nucleus of the cell. Next, the steroid hormone receptor complex binds to a specific segment or sequence on the DNA strand called the hormone response element. What I want you to appreciate here is that each steroid hormone receptor complex has a specific place to bind to on the DNA. It can't just bind anywhere on the DNA. This binding of the steroid hormone receptor complex to the hormone response element on the DNA initiates transcription. It basically causes the DNA to start pumping out large quantities of mRNA, and you know what happens next? The mRNA exits the nucleus to enter the cytoplasm where it attaches to a ribosome to initiate translation. So in other words, steroid hormone receptor complex stimulates protein synthesis of the target cell. Finally, thyroid hormone mechanism of action. Thyroid hormone mechanism of action is very similar to the steroid hormone mechanism of action. The lipid-soluble thyroid hormone diffuses through the target cell's plasma membrane to enter the cytoplasm, but it doesn't stop there. The thyroid hormone itself, continues into the nucleus. Inside the nucleus of the target cell, the thyroid hormone will bind directly to its specific receptor. The thyroid hormone receptor complex binds to a specific hormone response element on the DNA strand, just like the steroid hormone receptor complex, and stimulates the DNA to synthesize mRNA, again, transcription. And just like the steroid hormone mechanism of action, the mRNA will exit the nucleus to enter the cytoplasm where it attaches to a ribosome to initiate translation. In other words, both steroid hormones and thyroid hormones stimulate protein synthesis in their target cells.